This is a follow-up paper to the 2015 Oswald paper, where they discovered that glioblastoma cells can form tumor microtubes when implanted into mice. So in this paper, they tested various treatments. So before getting into the paper, I will do a glioma background again for like the third time. So glioma is a broad name for the general category of brain tumors originating from glial cells. So glial cells are the supporting structure of the neurons, and they help maintain the environment. So there's several types, and the one that we will focus on is the astrocytes. So astrocytes provide structure to the neuron. They can provide nutrients. They can also regulate the ion concentration, and they can also transmit calcium waves. So the reason why we focus on astrocytoma is because glioblastoma forms from astrocytes, and this type of cancer is highly invasive and it is hard to treat because the glioblastoma cells can form tumor microtubes. So tumor microtubes are thin, long protrusions extending from the cell body. They can extend up to 500 micrometers, and they help with a cell proliferation and a cell invasion. And TMs can also connect to each other through connexin 43 gap junctions, which help them transmit calcium waves. So there are several treatments for glioma. There's surgery that removes a part of a tumor. It is kind of impossible right now to remove the whole tumor due to the diffuse nature. There's also radiotherapy that provides a radiation dose to the patient. And then this radiation helps break down the tumor. There's chemotherapy, which injects a cytotoxic drug, or it's not a toxic drug to the tumor, and it helps break it down. There's targeted therapy, which targets specific molecules that help the cancer grow and spread, and it inhibits them, so it hinders the tumor growth. And then there's anti-inflammatory treatment, which prevents or helps prevent inflammation. So in their experiment of the paper, what they did is they took the two common glioblastoma strands, S24 and T269, and from human tumors, and they were grown in vitro, and then they were implanted into mice. The key difference between these two strands is that S24 tends to form more tumor microtubes in vivo. And then after implanting them into mice and growing them for some time, they tested various treatments. The first treatment that they tested was a surgical treatment. So in this image here, what we see is glioblastoma cells in green and the blood vessels in blue. So what they do is they take a syringe that has a 300 micrometer diameter, and then they remove part of a tumor. So this is before and then after. So they removed like this circle right here. And this will be referred to as the lesion from now on. And then they took a series of images after letting it grow back. And then we see it grows back on day 7 and then gets more denser in day 14. And then it gets really dense on days 21 and 23. So the authors note that the regrowth is actually faster and denser post-surgery than if you were not to perform this treatment. And this is the reason for this is due to tumor microtubes as well as wound healing mechanisms, hypothesized. This is the hypothesized reasons. So after doing a surgical treatment, they also took a closer look at what the cells are doing. So for these images, they're just looking at how the cells are moving. So the day before the surgery, they're tracking these 3 cells. After 100 minutes, they see that they haven't moved anywhere. So they're just diffusing. On day 5, however, post-surgery, they're tracking these 2 cells. And then on the next image after 100 minutes, the cells have moved to the right slightly. So they're seeing that the cells are actually moving towards the lesion. And then they do some quantitative analysis where the day before the surgery, the speed is lower than the speed after the surgery. And also, as days progress, more and more cells tend to move towards the lesion, as seen by the other bar graph. Now they also take a look at what the cells are doing. So they track this red cell here. After removing the lesion, they actually see that it forms tumor microtubes, and it actually puts them inside the lesion, and it can grow them outside. And then after tracking what the tumor microtubes are doing, they notice that the tumor microtubes tend to point towards more towards the lesion as days progress. So, yeah. Then they tested another treatment called targeted therapy. So in the top row is the control. So now in green are the blood vessels, and the glioblastoma cells are in red. For this treatment, what they do is they treat the tumor with hairpin RNA, which inhibits GAP43. So this protein is what helps tumor microtube formation. So after treating it, the cells are less likely to form tumor microtubes. So then, in comparison to the control, the tumor does indeed grow back slower. Then they also tested an anti-inflammatory treatment. So now this time the blood vessels are in blue and glioblastoma are in red. For this treatment, what they did is they took an anti-inflammatory drug, dexamethasone, and injected it on the day of the surgery, and then every day for 14 days post-surgery. And then they noticed that on day 7, the tumor is growing back slower, but then on day 14, there is no difference between the control and the surgery with an anti-inflammatory treatment. So it only had a short-term benefit. Now here is just a quick summary of their results. So they found that surgery causes the tumor to grow back faster and denser. Surgery plus targeted therapy significantly slows down the tumor growth. Surgery and an anti-inflammatory treatment only provide a short-term benefit. And then chemotherapy, I did not show the picture because they did not do it with surgery, but quickly here, chemotherapy significantly improves the survival time of the mice. And they find that the cells that have more tumor microtubes are more resistant to this type of treatment. Okay, so now on to the second part of this talk, which is the mathematical modeling of these experiments. So our modeling situation that we're going to try to model with equations is the following. So we simplify our domain to be symmetrical and to make it a circle. We will have a lesion in the center of radius 150 micrometers. So then we take the outer circle to be some radius that's greater than the one in the lesion. And then we will have cancer cells, and they will have preferential movement towards the lesion, but they can also move in other directions as well. So for their movement of the glioblastoma cells, we will assume that if they move towards the lesion, they are moving along the radial lines. And the closer they are to the lesion, the more preferential movement they have. Now, this situation, the particles moving along the radial lines have been previously modeled by Bika et al. So this thing is blocking, but it's okay. So what they did is they took a fully anisotropic diffusion equation given here, and they calculated the case for the radial movement in 2 dimensions. So they took a previously calculated diffusion tensor from the bimodal von Mises distribution, substituted into this equation, and then transformed it into the polar coordinates, which is given here. So this equation describes particle movement along radial lines. So we just have U derivatives in terms of space here, and we have this alpha R function. So this alpha R function describes the degree to which the particles move along radial lines. So the closer the value of alpha R is to 1, the more they tend to move along the radial lines. But if it's 0, then we fully recover the isotropic diffusion equation, so no preferential movement. So we will denote this whole equation with phi. So to model alpha R, we choose this equation with these parameters. And essentially what it does is describes the closer if the cells are closer to the lesion, alpha R grows, and the further away they are from the lesion, the closer it is to 0. So this is what that equation does. Now, to model the rest of the dynamics, we make a series of assumptions. So we assume that the healthy cells move through isotropic diffusion. We assume that healthy cells and healthy cancer cells and healthy cells grow logistically. We assume that the cancer cells grow faster and move faster than healthy cells. We also assume that the wound healing response causes the cells to grow faster and to a larger capacity in the lesion. And this is because when there's a wound healing mechanisms, growth factors are secreted, and also angiogenesis occurs, which provides more nutrients to the tissue. We assume that the tumor microtubes help repopulate the lesion. And finally, for the simplifying assumption is that the areas inside and outside the lesion are homogeneous. So we ignore any effects from the blood vessels. So this is our final mathematical modeling. U is going to denote the cancer cell density. V is going to denote the U denotes the cancer cell density, V denotes the healthy cell density. The first equation describes the cancer cell density, the rate of change of the cancer cell density with respect to time. So the first term is just the movement term, where phi was the term for the isotropic diffusion, and U is the diffusion coefficient. The second term is the growth term, where GU is the growth rate of the cancer and K is the carrying capacity. The second equation describes the rate of change of the healthy cells with respect to time. Now here, this movement term, they move isotropically. So this is just the diffusion equation in polar coordinates with a diffusion constant of dV. And the second term is the logistic growth, where GV is the growth rate of the healthy density. Okay, so these are the parameters that we use. So there is going to be 2 parameter values, one for inside the lesion and one outside the lesion. So recall, we have 2 regions. So this is inside the lesion, this is outside the lesion. So this is our parameter. So the diffusion is the same for the cancer cells. The diffusion here for the, sorry, this is cancer cells. This is healthy cells. So diffusion is higher for the healthy cells, since we assume that there's more movement, more space available inside the lesion to move. The growth rate for the cancer cells is higher within the lesion due to the benefit from tumor microtubes, as well as the wound healing mechanism. And the growth rate for the healthy cells is also higher within the lesion because of the wound healing mechanisms. Same with carrying capacity. So now to simulate the model, we need some initial conditions. So inside the lesion, we take the total total density of cancer cells and healthy cells to be 0. And for outside the lesion, we take the cancer cell density plus the healthy cell density to be equal to the carrying capacity. And we assume that the cancer cells make up 10% of that density. And then for the boundary conditions, we use the Neumann boundary conditions. So after setting all these parameters, this is what our simulations look like. So to get these, first, this is showing the cancer cell density. Just so the more blue it is, the lower the density is, and the more yellow it is, the higher the density is. So these were obtained in MATLAB through the PDP solver. So it's difficult to see, but there's a darker circle right here that says it's 0. and then outside it's slightly more blue. And then here there is regrowth, but you can't really see it. But then here we clearly see the regrowth, and then on the days we have it more and more yellow. So we are able to replicate the experimental trend where we see the regrowth where it's much higher and denser. And also we see that it invades into the nearby tissues as well, like we saw in experimentally. So just to see, this was the experimental. So we are kind of able to replicate it. Yeah. And ours are more symmetric than the experimental, and this could be due to the influence from the blood vessels that we ignored. Okay. So we perform sensitivity analysis on our parameters, and we find that the parameters that are most responsible for increasing the cancer cell density is the growth rate of the cancer and the carrying capacity. So from this, we know that the treatments that will be most effective are the ones that will lower those two parameters. So the first treatment that we try is surgery plus targeted therapy. So what we do for this treatment is we know that it inhibits the tumor microtube formation. So we assume that based on this, the cancer growth rate is lowered, as well as the degree of directed motion is lowered. So then what we do is we just lower the cancer growth rate by 50%, and we lower the upper bound of alpha R by 75%. And then we get these resulting images. So in the top is our control, and in the bottom is our simulated treatment. And we see that the regrowth is slower. So there's two color bars for these images because these ones have the first three columns have a lower concentration than the last one. So, and then the second treatment that we test is the anti-inflammatory treatment. So to simulate this, we didn't really have immune response data. So we assumed that the anti-inflammatory treatment loses its effectiveness over time. So this is due to the treatment not being permanent, as well as a potential resistance that's happening in the cells. So to simulate it, we assume that initially it works well and at delaying the immune response. So it's the growth rates of both cancer cells and healthy cells are not as extreme as we had in our base parameters, so we lower these. And then we also lower the carrying capacity because angiogenesis is delayed. And then we assume that the treatment starts losing its effectiveness after 3.5 days, and this was motivated by the data. And then these assumptions can be modeled by a sigmoidal curve or a hyperbolic tangent. Doesn't really matter, it will give similar results. And this is what we get. So the control is in top row and the treatment is in the bottom row. So then on day 7, it is lower than this one, but again, you can't really see. And then on day 14, it starts catching up to the original density. So it is still lower, but it's close to the one on day 14. And then on day 28, it is virtually the same. So it's not quite what is seen in the experiments, but it's a similar trend. All right, so that's everything. So we were able to come up with a fairly simple mathematical model that is able to capture the essential trends. Based on our model, we can hypothesize that tumor microtubes as well as the wound healing response are the main cause for the faster and denser regrowth of the tumor. We can conclude that the treatments that lowering the growth rate of cancer cells are the most effective. We've tested the assumption of if cancer cells move isotropic through isotropic diffusion. So here what we did is we had anisotropic diffusion, but if we assume that the cancer cells move through isotropic diffusion, then the results are comparable and there's not a lot of difference between the simulated densities. The model can also be extended to include competition between the healthy cells and cancer cells. We did that, and the results are as expected. The population that has a competitive advantage grows better. Finally, with more data, we can create a more accurate model. For example, if you had the immune response data, we could actually have the equations for the immune response. And finally, we did not consider the heterogeneity in the environment. Considering it could help us recreate the asymmetric tumors that we saw. And that's everything. Yeah.